So previously we have talked about the carbohydrates, in which we have talked about three types of carbohydrates. Can you tell me what are the three classes of carbohydrates? Monosaccharides, then
Disaccharide and polysaccharide.
disaccharides and polysaccharides. Can you tell me any example of monosaccharides?
Glucose,
Mm-hmm. Monosaccharides can we
fructose,
have trioses,
fructose,
hexoses, and pentoses, right? फर्स्ट ये आपके पास तीन ऑर्डर्स आते हैं. We have trioses, hexoses, pentoses. Hexoses की अब तीन टाइप्स बताएं. The one you just named.
fructose, glucose,
Galactose, right? Or glucose has two types. What are they? Alpha and beta. And we that also discussed that when two alpha glucose combine, they form disaccharides, such as in disaccharides, we talked about maltose. Then when you combine alpha glucose and fructose, we have what? Sucrose. And when we combine alpha glucose and galactose, we have lactose. Then we also discussed about some polysaccharides. In your syllabus, we have three of them. We have starch, glycogen, and cellulose. And starch has two types: amylose with 1,4 linkages and amylopectin with 1,4 and 1,6 linkages. Got it?
yeah.
This was everything that we studied in carbohydrates. Now, when we talk about lipids, lipids are very varied group of chemicals. They are all organic molecules which are insoluble in water, and most lipids are formed by fatty acids combining with an alcohol. The most familiar lipids are fats and oil, where fats are solid at room temperature and oils are liquid at room temperature. Now, lipids has two types. Lipids can exist as solids as liquids as well. If lipids are solids, then they are known as fats, and if they are in a liquid form, we call them oil. And chemically, they are all very similar. Fatty acids are a series of acids, some of which are found in the lipids, and they contain the acidic group COOH, known as the carboxyl group. The carboxyl group forms the head of the fatty acid molecule, and the common fatty acids have long hydrocarbon chains and everything. So fatty acids come in many types. We have long tails fatty acids as well, short chain fatty acids as well. And the name suggests hydrocarbon tails would only contain hydrogen and carbon. So the tails of fatty acids have double bonds between them as well. And we say that these fatty acids are unsaturated. Unsaturated means what? Double bond. Saturated tails means only single bond. They form unsaturated lipids. Double bonds make fatty acids melt more easily because the more unsaturated they are, the more double bonds they have, the more fluid they are, the more bends they would have in the chain. Due to these double bonds, तो उनकी fluidity would increase. For example, most oils are unsaturated and there is more than one double bond. The fatty acid or lipid is described as polyunsaturated. If there is only one, it is monounsaturated. If we have more than one double bond, polyunsaturated hydrocarbon tail, or if we have only one double bond, it's a monounsaturated hydrocarbon tail. Animal lipids, for example, are often saturated. They have no double bonds, whereas plant lipids are unsaturated. Animals have no double bonds. Plants do have double bonds. So we can say that animals, all of them have saturated tails, whereas plants have unsaturated tails. Just most of the oils that we occur from plants, including olive oil, sunflower oil, they all have unsaturated double bonds. Alcohols are a series of organic molecules which contain a hydroxyl group OH attached to a carbon. Glycerol is also a type of alcohol. Now, why are we studying about alcohols and fatty acid? Because a lipid molecule, remember, which we also call as a triglyceride, is always made up of one glycerol and three fatty acid chains. This is something that you already remember from your IGCSE as well. So triglyceride structure का नाम होता है that we are going to study. But before that, we are just taking a brief about this glycerol and the three fatty acid chains. So glycerol, as I am just talking about, is also an alcohol. It has an OH attached to a carbon atom, and the reaction between an acid and the alcohol produces a chemical known as ester. The chemical link is also known as the ester bond. And how does an ester bond form when an acid reacts with alcohol? Alcohol loses an H and the acid loses an OH. When this water is removed, there is an ester bond formed. This is what we call as an ester bond, and the water is a byproduct. This ester linkage is a chemical bond represented as COO formed when an acid reacts with an alcohol. So here you can see this is a saturated fatty acid with all single bonds, but this is an unsaturated fatty acid, okay, which has double bond. Look at this, one double bond, the second double bond, third double bond, and this double bond, if you look at both of the tails, this is a relatively straight chain. But if you look at an unsaturated chain, it is often having these bends in it as well. And these bends basically provide lipids fluidity. Got it?
Yes.
So how is a triglyceride formed? One glycerol molecule combines with one, two, three fatty acid chains, forming in ester bonds. This is what we call as a structure of a triglyceride. Ide is due to oxygen, glycer is due to the glycerol alcohol, and tri means three fatty acid chains. One, two, three fatty acid chains combined with one molecule of this green glycerol. This is what a molecule actually looks like when you combine them. Glycerol combining with three fatty acid molecules, a condensation reaction where H2O would be removed, like I just explained above, and triglyceride will be formed. The COOH group of the acid reacts with the OH group on the alcohol to form the ester bond. This is a condensation reaction because water is formed as a product. The resulting ester can be converted back to an acid and alcohol by the reverse reaction of adding water. It is a reaction known as hydrolysis. The most common lipids are triglycerides. These are the fats and oils. A glyceride is an ester formed by a fatty acid combining with an alcohol glycerol. And as you have seen, glycerol has three hydroxyl groups. Each one is also able to go on a condensation reaction with a fatty acid. And when we have seen when this triglyceride is made, the final molecule contains three fatty acid chains and three ester bonds. The tails can vary in length depending on the fatty acids used as well. Triglycerides, you should know there's some properties as well. Triglycerides are insoluble in water, but soluble in organic solvents such as ethanol because they themselves contain an alcohol glycerol as well, but they are not soluble in water. Have you ever seen that oils dissolve in water? Oil always becomes a layer on top of the water. It never becomes soluble because if we talk about a triglyceride molecule has two parts. No? One of them is a glycerol part and the other part is the fatty acid chains part. The glycerol part is polar, but the fatty acids part, the three fatty acid chains, they are non-polar. Now, because of them being non-polar, they are hydrophobic. Hydrophobic mean they hate water. They have a phobia from water. And glycerol head is polar, which means it's soluble because it is hydrophilic. It is what? Hydrophilic. Hydrophilic means it is water loving. So only the heads go to the water, but the fatty acids stay pointing out of the water. The fatty acids never dissolve. So overall, the whole triglyceride molecule doesn't dissolve as well. This glycerol is polar, hydrophilic, and soluble. It loves water. But the three fatty acid chains, they are non-polar, insoluble, and hydrophobic by nature. Consequently, they are hydrophobic and they do not mix freely with the water molecules as you have seen in the diagram above as well. So what is a triglyceride? It is a type of lipid formed when three fatty acid molecules combine with glycerol and alcohol with three hydroxyl groups. Now, what are the functions of triglycerides? Can you tell me? Any idea what are the functions of these triglycerides?
Insulation,
What else?
maintaining heat, maintaining temperature,
Mm-hmm.
storing fat,
Yes.
um, and for example, myelin sheath.
Mm-hmm. Good job.
Okay, now this is the first video looking at lipids, and lipids is a scientific name for molecules found in fats and oils. Lipids have a range of functions, and you need to be able to describe them. Firstly, lipids are a major source of energy in the human diet. We find lipids in oils such as olive oil, and in solid fats such as butter. Both of these are rich sources of energy. Lipids are also used to store energy, for example adipose tissue in humans. adipose tissue is found under the skin. Now, as well as acting as an energy store, this adipose tissue also helps to insulate the body, reducing heat loss to the environment. We also find adipose tissue around internal organs such as the kidneys, and this helps to protect these organs from injury. Lipids are also used as waterproofing, for example the oils which coat the feathers of aquatic birds. And finally, lipids are a major part of the structure of membranes, such as those which surround cells and mitochondria. Now, there are two main categories of lipids. These are called triglycerides and phospholipids. In this video, we're going to look at triglycerides, and in the next video, we look at phospholipids. This shows the general structure of a triglyceride. Triglycerides contain a molecule of glycerol bonded to 3 fatty acid molecules. So let's start by looking at fatty acids. I'm showing you the structure of a fatty acid molecule here. Now, there are two key parts to fatty acid molecules. At the end, we have the carboxylic group, and this plays an important role when we form a triglyceride. We'll be looking at that in more detail later in the video. The rest of the molecule consists of a long chain of carbon atoms bonded to hydrogen. Now, this is a saturated fatty acid. Saturated fatty acids contain only single covalent bonds between the carbon atoms, and we can see those here. I'm showing you here an unsaturated fatty acid. As you can see, unsaturated fatty acids have at least one double covalent bond between the carbon atoms. This is a monounsaturated fatty acid, as there's only one double covalent bond in the carbon chain. This fatty acid has 2 double covalent bonds in the carbon chain, so this fatty acid is polyunsaturated. Coming up, we're going to look at the formation of triglycerides. Okay, now as we saw before, triglycerides are formed from 1 molecule of glycerol and 3 molecules of fatty acids. I'm showing you a molecule of glycerol here, and I would recommend that you learn this structure. As you can see, a glycerol molecule has 3 hydroxyl groups. Here's a glycerol molecule and a molecule of fatty acid. Now these two molecules can react together to form a bond, and I'm showing you that here. This is called an ester bond, and this process is called esterification. In this reaction, we also make a molecule of water, so this is an example of a condensation reaction. Now we can carry out this reaction with 2 more fatty acid molecules like this, and this is the structure of a triglyceride molecule. Now, as we said before, fats and oils are a major source of energy in the human diet. In the digestive system, lipase enzymes break the ester bonds, releasing the glycerol and fatty acid molecules. This reaction requires 3 water molecules and is an example of a hydrolysis reaction. Now, there are a couple of final points about triglycerides that you need to understand. Firstly, triglycerides are non-polar molecules. This means that they're hydrophobic. In other words, they do not dissolve in water. This explains why triglycerides are used as waterproofing, for example in aquatic birds. Secondly, because triglycerides contain a large number of carbon and hydrogen atoms, a great deal of energy can be released from triglycerides, and we'll see that in more detail in the topic on respiration. In the next video, we look at the category of lipids called phospholipids.
Alright, is the structure of triglycerides clear?
Yeah.
ठीक है, clear हो गया? Done?
Yes.
So as you mentioned, we have many functional triglyceride forming the myelin sheet, providing insulation. So triglycerides, first of all, they are excellent energy stores. Why do you think they are excellent energy stores?
Because they are insoluble.
No. Because they have high number of carbon-hydrogen bonds. And when you are going to break these hydrogen-carbon bonds, energy would be released. They have more carbon-hydrogen bonds, even more than carbohydrates. A given mass of triglyceride will therefore yield more energy on oxidation than the same mass of carbohydrate. It has a higher calorific value, an important advantage that it has for being a storage product as well. Triglycerides are stored in a number of places in the human body, particularly just below the skin and around the kidneys. Below the skin, they also act as an insulator against loss of heat. So first use and function was providing energy. Second is that they provide insulation against loss of heat. Blubber, a triglyceride found in sea mammals such as whales, has a familiar function as well as providing buoyancy as well. An unusual buoyancy is also floating. They have a role in making the seals float on water. An unusual role for triglycerides is a metabolic source of water. Remember, fats are also source of water. Even if there are two people who are very thirsty, the fat person would have still some stores of fat that can be converted into water. That is known as fat burning. When these fat stores are oxidized in respiration, triglycerides are converted to carbon dioxide in water. How does a person lose fat? When he has already burned all of the glucose in his body in respiration, now comes the turn for fat. That fat is now burned in respiration to produce carbon dioxide and water. The water may be of very importance in very dry habitats as well. For example, the desert kangaroo rat never drinks water and only survives on the metabolic water from the triglyceride containing the food it actually eats. Got it?
Yes.
Now let's talk about phospholipid molecule. What are phospholipids? By the name, I can tell that it has a phosphate group, and lipid means that it also has a glycerol and fatty acid chains. Glycerol and fatty acid chains are lipids, but now they are combined with phosphate. They are a special type of lipid. Each molecule has the unusual property of having one end which is soluble in water, and this is because one of the three fatty acid molecules is now replaced by a phosphate group, which is polar and can now dissolve in water. The phosphate group is hydrophilic and makes the head of the phospholipid molecule hydrophilic. How? First, the lipid used to be insoluble when it had one glycerol molecule and three fatty acid chains, right? But now what is wrong with phospholipid? Phospholipid is a molecule which replaces one of the fatty acid chains and attaches itself over here. Now this molecule is known as a phospholipid. Why? Because now this molecule has one glycerol, two fatty acid chains, and one phosphate molecule. And this phosphate molecule is polar as well. It is soluble as well. And it is hydrophilic, water loving. That is why in chapter number 4, you will see, and you have already seen as well, that phospholipids make up the cell surface membrane of the molecule. Now, there are still two remaining hydrocarbon tails which are still hydrophobic. You have seen now, now our new phospholipid molecule is how? It has one glycerol, one phosphate, and two fatty acid hydrocarbon tails. So this part is still what? Insoluble in water. But this part is soluble. This allows the phospholipids to form a membrane around a cell that you'll see in much more detail when we talk about chapter number 4, where the phospholipids are arranged with their hydrophilic heads in water solutions and so on. No need to talk about that now, Beshak. It's a part of your chapter 4. Let me show you one more thing. So can you tell me the structure of phospholipids?
One phosphate, one glycerol, two fatty acid chains.
Good job.
Hi and welcome back to Free Science Lessons. By the end of this video you should be able to describe the structure and function of phospholipids, and if you're following the OCR spec you should be able to describe the role of cholesterol. In the last video we looked at triglycerides. We saw that a triglyceride molecule contains one molecule of glycerol bonded to three fatty acid molecules. Now one key fact about triglycerides is that they're non-polar. In other words, the molecule is uncharged. This means that triglycerides are hydrophobic. In other words, they will not dissolve in water. This makes triglycerides useful for waterproofing, and we saw that in the previous video. Now, phospholipids have a structure which is similar to triglycerides but different in one key way, and this difference has a major effect on the way that phospholipids behave. I'm showing you the structure of a phospholipid here. As you can see, a phospholipid has a glycerol molecule bonded to two fatty acid molecules. The glycerol molecule is also bonded to phosphate. Now, phosphate looks complicated, but you don't have to learn the structure. The key idea that you need to get is that the phosphate is negatively charged. In other words, this part of the molecule is polar. Because of this, the phosphate group is hydrophilic. In other words, it attracts water molecules. Now, I should just point out that in reality, phospholipid molecules are slightly more complicated than this, and that's because the phosphate can attach to other groups such as sugars. We look at that in more detail in later topics. I'm showing you here a simpler way to represent a phospholipid molecule. In this case, the head group is the polar hydrophilic part of the molecule. The tails represent the two fatty acid molecules, and remember that these are non-polar and hydrophobic. So as you can see, phospholipids contain both a hydrophilic and a hydrophobic region. This means that phospholipids behave very differently in water compared to triglycerides. In water, phospholipid molecules position themselves so that the hydrophilic head groups can interact with the water molecules, while the hydrophobic tails cluster together well away from water molecules. Scientists call this structure a phospholipid bilayer. Now, this property of phospholipids is extremely useful as it allows phospholipids to form the membranes that we find both around cells and within cells. You'll be seeing this again when we look at cell membranes in more detail in later videos. Coming up, we're going to look at cholesterol. Okay, now if you're following the OCR spec, then you need to be able to describe the properties and functions of cholesterol. Cholesterol is part of a family of lipids called sterols. As you can see, cholesterol does not look anything like triglycerides or phospholipids, and I should point out that you're not required to learn the structure of cholesterol. Now, there are two key features of the cholesterol molecule. The hydroxyl group is hydrophilic. However, the rest of the molecule is hydrophobic. Now, what this means is that cholesterol can insert into cell membranes. The hydrophilic hydroxyl group on the cholesterol molecule can interact with the head groups of phospholipids, while the rest of the cholesterol molecule can interact with the hydrophobic fatty acid tails. Cholesterol actually plays a key role in controlling the fluidity of cell membranes, and we'll see that in more detail in the cell membranes topic. Now, cholesterol plays a number of other critical roles in living organisms. Firstly, cholesterol is used as a starting point for a range of hormones, including estrogen and testosterone. Because they're based on cholesterol, these hormones can pass through cell membranes and interact with their receptors inside the cell. Secondly, cholesterol is used in the body to make vitamin D. This takes place in the skin in response to ultraviolet light. Vitamin D is needed for the proper development of bones. And lastly, cholesterol is used in the liver to produce bile. The function of bile is to increase the rate of digestion of lipids by the enzyme lipase. Okay, so hopefully now you can describe the properties and functions of phospholipids and cholesterol.
Alright, phospholipids का भी समझ आ गया?
Yes.
ठीक है. So let me show you one thing. उसके बाद कुछ past papers questions भी we'll do them as well. If we look at the summary regarding the first two parts, if we talk about the carbohydrates and lipids, so remember, number one, the larger biological molecules are made from smaller molecules. These larger biological molecules can also be said as polymers, which are made up of their monomers. The smaller molecules are joined together by condensation reaction by the loss of water, where the condensation involves removal of water, and the reverse process, adding water, is called hydrolysis, which is used to break the large molecules back down into smaller molecules. So hydrolysis means adding water, where you can break polymers back into monomers, and condensation is removal of water, where you join monomers to form the polymers. Condensation is done by loss of water. Hydrolysis is done by addition of water. Hydrolysis means breaking. Condensation means joining. Polysaccharides are made from monosaccharides, proteins, amino acids, lipids, fatty acids, glycerol, and polysaccharides are also those proteins which are formed from identical or similar subunits called monomers. They are therefore known as polymers as well, क्योंकि वो भी macromolecule बना सकते हैं. Carbohydrates, on the other hand, have a general formula of CxH2OY, which means carbon and oxygen would always be in the same ratio, but hydrogens would always be double. For example, glucose is C6H12O6. Even if it includes your monosaccharides, disaccharides, and polysaccharides, the ratio would always be the same while writing the formula. Monosaccharides are joined together by glycosidic bond, as we saw when glucose and glucose combine to form maltose. They are all formed by glycosidic bonds. When glucose joins with fructose, By a glycosidic bond, they form a disaccharide, sucrose, which are all very water soluble and are known as sugars. They are also an important energy source in cells and also important building blocks for larger molecules like polysaccharides. Monosaccharides may be straight chain or ring structures, and they may exist in different isomeric forms, such as glucose has two isomers, alpha glucose and beta glucose. In alpha glucose, the OH is below the plane, and in beta glucose, the carbon number 4 OH is above the plane. Benedict's reagent can be used to test for the reducing and non-reducing sugars. Reducing sugars may you have monosaccharides and disaccharides. Non-reducing sugars may you have your polysaccharides, which do not show the color change. Polysaccharides include starch, glycogen, and cellulose. Starch has two types, okay, which are amylose and amylopectin. Starch is the storage molecule in plants. Glycogen is the storage molecule in animals. And where is cellulose found? Can you tell me?
Plant cell wall.
Plant cell wall. Starch is also an energy storage compound in plants, and glycogen is the energy storage plant in animals. Because both of them produce glucose. How? Plants make their glucose by photosynthesis. They store their glucose in the form of starch. We obtain glucose from plants and we store our glucose in the form of glycogen. Iodine solution is the one that is used to test for starch. Iodine solution has a color which is orange brown, and when you add it to starch molecule, it will turn into blue-black. Blue-black color is a positive test that will prove that yes, starch is present over here. Glycogen is an energy storage compound in animals. It is made from alpha glucose and its structure is similar to that of amylopectin, but with more branching. Cellulose is a polymer of beta glucose molecules, and the molecules are grouped together by hydrogen bonding to form mechanically strong fibers with a high tensile strength. Yes, cellulose ki we talked about, these are different fibers that when joined in a crisscross arrangement, they form a very strong molecule, even harder than steel, which are found in plant cell walls. Lipids, as we have just discussed in today's lecture, lipids are a diverse group of chemicals, the most common of them which are triglycerides, made up of one glycerol molecule and three fatty acid chain. Triglycerides are made by condensation between three fatty acid molecules and glycerol, where ester bonds join to the fatty acids to the glycerol. They are all hydrophobic, they do not mix with water, and they act as storage compounds. They also provide insulation, buoyancy in marine animals. But when we talk about phospholipids, they have a hydrophilic phosphate head and two hydrophobic. अब ये जब phospholipids आ जाते हैं ना with their phosphate head, now it is soluble. But when we had glycerol and three fatty acid chains, they were insoluble. Now in phospholipids, we have a glycerol, one phosphate, and two fatty acid chains. This is soluble now. And emulsion test is used for lipids where you use ethanol. Ethanol use karte hain, and if a white cloudy emulsion is produced, that is a test for lipids. There's a whole separate topic that we'll discuss for food tests as well. Is the summary clear?
Yes.
Hi guys, in this video we'll look at an introduction to lipids, the proteins and carbohydrates. And lipids come in two main types that we find in the body. There are triglycerides and there are phospholipids. We'll go through the detailed structure of each of these, but a triglyceride normally looks like this, with one molecule here and then three parts coming off at the end. And the other type is called a phospholipid, with its distinguishing feature being this phosphate group lying on the end. Because there are two types of lipid, the triglycerides and the phospholipids have different but both important roles. Triglycerides are more used as a source of energy. We can find them in a variety of foods that we eat, for example oily foods, and butter, and certain fats as well. And oils found in various plants too, and they're used to provide us with energy. On the other hand, phospholipids aren't used for energy so much, they're used more for a structural role in building up the cell membranes. And the membranes that surround the cell act as the kind of barrier to the outside world. And the main component making up this layer is called a phospholipid. So lipids themselves, whether we're talking about triglycerides or phospholipids, are mainly composed of the elements carbon, hydrogen, and there is some oxygen thrown in there as well. So it mainly has the same elements as carbohydrates. So we can see that there's lots of carbon across the chain of the molecule, there's evidence of hydrogen, and there's some oxygen as well, although the oxygen is a lot less common. We tend to see this long chain of carbons and hydrogens and this is known as a hydrocarbon chain. So hydrocarbon chains are often found in these lipid structures, and at the end we have different groups depending on the different lipids. They're very large complex molecules, so we call them macromolecules, with macro meaning large, but they are not polymers. So when we talk about polysaccharides, or proteins, and nucleic acids, we tend to say that they're polymers made up of multiple units known as monomers. But in this case, for lipids, they're not polymers because each of the units is not a monomer. It's simply a long complex molecule, but it doesn't have individual units you can break it down into. We also describe lipids as being non-polar molecules, and so if we were to add them to water, they will not dissolve in the water. So polar molecules are able to dissolve in the water and mix into the water to make a solution. If they're non-polar, then they're unable to make any connection with the water molecules in water. So the oil and the water never actually mix. You can sort of swirl it around and get bubbles, but they will eventually form two separate layers as they want to stay well away from each other. So let's talk about the molecular structure of triglycerides. And remember, these are the types of lipids we use to give ourselves a source of energy. A triglyceride molecule is formed from one molecule of a compound called glycerol, and it's connected to 3 fatty acids. So you can look at the name and work this out, because tri means 3, and the glyceride refers to the glycerol. So there's 3 fatty acids for a glycerol molecule. So looking at this triglyceride molecule, the red structure represents a glycerol, just one molecule, and these 3 chains represent 3 fatty acids. And these look very much like the long carbon chains we saw in the previous slide. So let's talk about each of these components. The glycerol is an organic alcohol, and it has 3 hydroxyl groups, or OH groups, on its side. So an alcohol is any time where you have a carbon chain which has these OH groups attached to it. And glycerol in particular is 3 carbons long, 1, 2, 3, and therefore it has 3 hydroxyl groups along it. So this is the glycerol. The fatty acids, and remember there are 3 of them for this triglyceride, are organic acids, so they're able to lower the pH of things. They have a carboxyl group, or a COOH group, on one end, and that end is joined to a hydrocarbon tail. So it's called, in general, a carboxylic acid. So let's break this down on the molecule. We've got this long chain of carbons and hydrogens. This is the hydrocarbon tail, made purely of carbons and hydrogens joined up. Now you might think this looks like a polymer, but it's not, because it doesn't have the same units repeating itself. We have this end which is different. Here we have this end which is the carboxyl group, and a carboxyl group always has a carbon, joins to one oxygen, and then another oxygen and an OH. So it's COOH. So the COOH group is joined to the hydrocarbon tail, and overall this is acidic and it's called a carboxylic acid. Often we don't tend to draw out the whole hydrocarbon chain, we use the letter R to represent the hydrocarbon chain. So this is exactly the same thing as the one above, only this time we've got the carboxyl group on the end as before, but we've turned the hydrocarbon tail, instead of being infinitely long and full of detail, we've just decided to name it R. This saves space and time in the long run. So looking at the molecule in more detail, we need to talk about the ester bond. So the triglyceride molecule is formed when we join one molecule of glycerol to those 3 fatty acids, and we do this through condensation reactions. So remember, the triglyceride has those 2 components. We have the glycerol joined to 3 fatty acids, and as these get joined, they form the triglyceride. And this type of reaction is known as condensation. Condensation is seen in lots of reactions in the body where we're joining things together. In a condensation reaction, it forms water, because if you think about condensation on the side of a glass bottle or on the mirror, it's where water droplets begin to form. So the reaction itself forms the water because 1 oxygen atom and 2 hydrogen atoms get removed from the glycerol and fatty acid. So let's take that in forms of a diagram. Here we've got our glycerol molecule, and then we've got one of these fatty acids approaching the glycerol, and remember the R represents this long chain of hydrocarbons. So in order to join the fatty acid to the glycerol, and remember this will happen 3 times, we need to remove certain atoms to make way for this. And what we do is we remove an H and an OH from both of these groups. So overall, we've got 2 H's removed and an O, so we form H2O. and this goes off and leaves, and obviously this is a molecule of water. So in condensation, two things are joined with the removal of water. And the particular bond that's formed between the glycerol and the fatty acid is what we call an ester bond. So now if we look at this structure, the glycerol has now got this connection with the fatty acid, and this is called the ester bond. And remember, the molecule of water which was formed has now left as a product. So what we have here is we have glycerol joined to one fatty acid, and we call this a monoglyceride. And hopefully you can see that with three of these we would have a triglyceride. Two would obviously be a diglyceride. So by definition, an ester bond is the covalent bond formed by a condensation reaction between the OH group of a carboxylic acid and the OH group of an alcohol. So looking back up, we used the OH group of the alcohol, which was on the glycerol, and the H or the OH on the fatty acid, and now they're formed together to make a covalent bond, which is this ester bond. So in order to make the triglyceride, we need three condensation reactions, forming three molecules of water. So here again we have our total glycerol molecule, and then on the right here we have three individual fatty acids, also known as carboxylic acids, and you can see this carboxyl group for each of them, and the R representing a chain of carbons and hydrogens. And every time we want to make one of these ester bonds, we remove water for each molecule coming together. So overall we form three water molecules, and we'll have a triglyceride with three ester bonds. And therefore the end product as a triglyceride has three ester bonds connecting one glycerol molecule to three fatty acids. And there are three. In forming ester bonds, we used a condensation reaction, and breaking them down, we used something called a hydrolysis reaction. So the triglycerides can be broken down into glycerol and three fatty acids, which are the components which make them up, in three hydrolysis reactions.
So in three hydrolysis reaction, आप एक एक बॉन्ड तोड़ सकते हो ग्लिसरी और यहाँ से आपके पास हाइड्रोलिसिस के थ्रू एनर्जी भी रिलीज हो जाती है. And that is the end of your lipids.